firstly, I would like to thank organizing committee for giving me the opportunity. I thank President Kelly University and Vice Chancellor and Professor Jagannath Rao sir, who is a mentor for me, and the France team for giving me this opportunity to present a few questions on drug discovery of on Alzheimer's disease. As I'll take few minutes just to quote what's still yet to be addressed in case of neurodegenerative diseases. You can see that, so we know that amyloid, tau, and synuclein has been widely known, but still we have few aggregates like AACD which has been there as aggregates of the brain. So we know that every person is different and their brain health can take different paths. So it's a scary statistics across the world, the global impact, as you know, by 2050, so it may be around 131.5 million by 2050. It is scary statistics. And of course, in India, this is the statistics of Indian statistics of neurodegenerative diseases, including Alzheimer's and the Parkinson's. You can see several states have been severely affected, etc. The data is very scary. So this program is very well organized at the right time. So these are the pathological events which we can see across this thing. Formation of A beta, we know, formation of tau, etc. And of course, all these things are pathological things which can be formed inside the neuronal cell. Not only that, this is a new thing. Everybody talks about A beta and tau, but we in our lab, we started looking at these AACD, which is one more pathological hallmark. Maybe we call that as third hallmark of Alzheimer's disease. So everybody talks about tau and A beta, but AACD is also equally problematic across the neuronal death. So there was no model. We have modeled the same here, and it is known that this can also bind to the DNA, which leads to the neuronal death also. So a lot of pathological symptoms has been seen, and of course you can see in India, we can see that state-wise, we can see that a lot of changes in the neurological disorders. So it's the right time to awaken. So we all know that these AD and PD, and especially in AD, you can see the progression of Alzheimer's disease here. So even approximately mild to moderate, you can see, and later it spreads across the brain here. We all know all these things. My question is that we are all looking in a different angle. So I have to pose 2-3 questions here. Like, these are all the mutations which has been identified across APP, which is a protein which gives rise to amyloid 1 A beta. And of course, we have done some of these mutation analysis, like genome-wide analysis of A beta protein. So if we look at the drug discovery perspective, we can see that many mutations can lead to issues. Like across the world, you can see Arctic, Dutch, and these are the mutation across the 1 to 42 residue. So we have to look at all these mutations very seriously here. So likewise, if you look at these issues like peptide assemblies and amyloid diseases, it's very important to understand for us that, so when there was an initial A beta was isolated, this was the thing, and later recent structures which were isolated from the patient exhibit these structures. So it's very, very important to understand these things. So you can see the 1C, this structure is a recently isolated structure from the Alzheimer's patient. So we have to understand these monomers to dimers in this case here. So now this is what one more challenge in front of all of us, that they have in intermolecular and intramolecular interactions. It has to be very well addressed nowadays, because when the monomer comes out, they start aggregating by bonding with either with the salt bridges, etc. So these are the challenges. You can see these are the lot of mutations which has been observed across the A beta here. Not only that, we have to look at the pathways which can affect or lead to the Alzheimer's diseases. So these are some of the kinase and phosphorylation pathways which has been observed across the Alzheimer's. So we can also look at these pathways and their drug targets as kinase and phosphatases. And not only A beta and tau or others affect the Alzheimer's disease, but there are cross pathway analysis has been observed over here. So there are a lot of genes which has been known to cause Alzheimer's disease, like starting from apoptosis to cell cycle P53 signaling pathways has been done, which are known to be affecting diseases. Likewise, we have mutations at tau. So these mutations are very challenging, which has to be addressed. And of course, all the kinases and phosphatases which has been there has to be analyzed for further. Because inhibiting the kinases is a challenging thing, but in Alzheimer's, we can see that these, there are a lot of kinases are involved in phosphorylating tau and leading to Alzheimer's diseases. So you can see the picture is very scary that these are all the mutations which has been observed across the tau protein here. So these many structures are available. How to target these structures is a big question mark now. So many structures isolated from the patient and which has some pathological signatures. So how to address these things is a big question now. Like this alpha-synuclein, all of us were talking about synuclein from past 2 days, but you can see the scary statistics of mutations which occurs across the synuclein, which has to be addressed here. So this is a challenge in front of all of us. You can see that the table gives us a lot of mutations which are being there, and tackling these things towards drug discovery is very, very important here. So this is what, this is how we can see that different polymorphs takes up inside the human brain. Pointer. Okay. So you can see that the formation of different polymorphs, this is challenging for drug discovery here. You can see. So given a patient, we can get any polymorphs in that individual. So how to target these synucleins is a difficult task. So these are some of the polymorphs of the tau. So it's very important to understand all these things. So these are the protein-protein interactions which has been observed across, intermolecular interactions and the mutations which has been observed, which are leading to toxicity of these peptides here. Lastly, is it possible for us to understand the pathological mechanism? You can see that AD and PD, even though they are different clinical and pathological features, but they share common pathological mechanism. So through drug discovery, is it possible for us to find out a common drug target for AD, PD, and HD so that we can, it's like 3 neurological disorders, 1 drug. So this is an interesting, pathway which we can think of targeting these neurological disorders here. So last few slides. Now our theme of the conference is artificial intelligence, machine learning and deep learning and virtual screening. So these are the probable drug discovery approaches where we can think of how we can use AI ML in case of drug discovery towards neurological disorder. You can see here in the first picture, we can see that lot of AI tools can be applied to understand this. The right side of this also we can see artificial intelligence algorithm can help us in speeding up the drug discovery for looking at various candidates, molecules. Like these are the recent ones. So big data, machine learning and deep learning, finally we can look at the neurological perspective. So recently a lot of reviews has come, and this is how AI can help us in in looking at the neurological disorders. AI-driven target discovery for neurological disorders. A lot of algorithms has been developed towards this here, and probably these are the slides where computer science professors can help biologists in solving these things. You can see a lot of machine learning algorithms, especially for nerve diseases, has been looked into. These are some of the tree illustrations which has been there. And of course, this is the deep learning methods which all of us can look into to understand and solve these degenerative diseases. So this is the final slide, like where future research direction in degenerative nerve diseases, where we can see that all these things can help us to understand and solve some of the problems. This last slide of mine, like this is the 1 week back thing which happened. There's a new initiative launched to accelerate use of AI in fight against neurodegenerative diseases, that is 10,000 Brain Project. So this is initiated by the Milken Institute Science Philanthropy Accelerator to develop a roadmap for the rapid adaptation of AI within the neurodegeneration ecosystem. I feel that people are looking at AI into neurodegeneration. This is 1 week back it was launched, and I feel personally that, and we are all here in Vijayawada and also thinking towards the same thing. It's a wonderful thing. This project may be very helpful for all of us to look back to neurodegenerative. So that's all. Thank you all. Thank you for the opportunity, sir. This also, even sir is my mentor, right? Jagannath Rao sir, he has been my guru. So these are some of the collaborative, collaborators for my work. Thank you one and all.
Long live India and France friendship.